Thank you, Maggie and Wendy, and thank you everyone for coming. So, we're going to talk about iNaturalist and the City Nature Challenge, and then be able to answer questions and show some of the website features and chat until everyone wants to sign off and eat dinner. So we're going to start with the broad question of what is community science or citizen science. The terms are roughly interchangeable, and this applies to the idea of iNaturalist and other platforms such as eBird and um, maybe the Xerces Bumblebee Watch. If you attended a presentation last month by one of the Xerces folks, talked about the Bumblebee Atlas, the Bumblebee Watch. So anything where people who are not paid entomologists or paid botanists are contributing data is roughly what we're looking at here. I copied definition from Wikipedia, but the general idea here is about accessibility and about making it convenient for lots of people to participate, regardless of your background knowledge, skill, and interest. And we're going to start with the City Nature Challenge that Maggie mentioned. So it's coming up at the end of the month. I have some more details on the next slides. And it's a competition in the sense that different cities and metropolitan areas and counties are competing, in some ways competing to have the most observations and the most species. But it's also this idea of a bio blitz of getting lots of people out there to try and find lots of things around the same time of year in multiple years, and maybe being able to produce some interesting data out of that. So it's been going on for a few years now, and iNaturalist is the host platform for the City Nature Challenge. It's where you have to upload things for them to count as part of this effort. So these are the dates that you want to know, and there was more information on the blog part of the iNaturalist website. So you have Friday, Saturday, Sunday, Monday, I think, to make observations, and then the following week to upload any pictures and make any identifications and then help others. So if you have any more questions about City Nature Challenge, we'll come back to those. In the meantime, we're going to talk about the iNaturalist platform and what makes it, say, better or different than other citizen science platforms. And one of the biggest things is, as it's become more popular and grown, you have a lot of users around the world. So you don't have, it's not just the fact that you have a lot of people taking photos and being able to discover interesting things where scientists don't necessarily have the time to visit all these places, but it also attracts experts in different fields who want to come see what those photos are like and help identify them and help improve the data. So this was a snapshot I took about a month ago when I was putting this together, and this is just for Chaffee County, and you can see about almost 2,500 people have uploaded at least one observation in the county, and almost 2,000 people have identified at least one observation in the county. And these are the top species that are frequently posted from the area. So pretty common stuff. In the second row, you've got your Hippomopsis and another penstemon, mountain mahogany. Well, and Spanish dagger, your common yucca and bee plant. So these are the things that people tend to see, maybe because of flowers or maybe just because they're common, like the pinions. And then if you were to upload something, these are the top three identifiers for the county. So I snipped Rick's bio from the website. So if you upload a plant in the county, um, he's identified at least 4,700 as of last month. And you can see that he's not just some random Joe off the street or random Rick, but he's a professional RMBL botanist. So one of the features of iNaturalist is you have all these other observations. And so if you want to go find something or you want to see, for example, where are bighorn sheep and where are my best chances of going to see one. So you can see that the points pretty much align with the highways, which makes sense. That's probably where people are seeing them, not necessarily where they're the most common, but the backside of Browns Canyon National Monument, perhaps, is a good spot along the road there, and through the actual canyon and along the river. So it's with sheep moving around, maybe this wasn't the best example, but just an idea of the kind of searches you can do. So because there's all these people on there, if you upload something, and I'll explain more about what this particular context is, you can ask people questions, and this way it's a bit like social media. You can tag someone and they'll get a notification about your message. So I found a wasp, a spider wasp at my house in Albuquerque, and I uploaded a photo of it, and then I tagged three of the top identifiers, asking for help. And then I also went and looked at another website. If you're trying to identify any kind of wasp or spider or butterfly, and arthropods in general, this website Bug Guide is also very useful. So it's a bit of a free internet guide. People work to identify images and move them around and add information. And so if you have an idea of something, for example, if you get, and I'll talk more about this, if you get the computer vision suggestion from iNaturalist, which is a piece of software, they have an algorithm that analyzes your photo and compares it to other photos. So if it spits out some name and of a wasp, for example, you could come to Bug Guide and then look at photos that are already identified of other wasps and see if they're similar. So one of the people, right, just going back, so one of the three people in this case, Sasha 151, I believe a Russian entomologist, responded to my question um, and said, okay, looking at your photos and I did some other internet research that they thought these were the characteristics that were important, and they said based on your photos, it's probably this species, Thoracicus. So that's one way that collaboration can work online, and I can show you that example in real life in the browser, so it's a little less disconnected, but it doesn't quite fit well vertically in a presentation screen. So this is what I was just talking about, the computer vision algorithm. So iNaturalist has their own model, their own programmers, and they take all the photos on the website and they feed them into the model with their identifications, and then it's a machine learning. I can't explain it, but things happen and it's used to predict the identifications of future photos. So this is iNaturalist's explanation of how the tool works, and I'll let you read that. And when you use it on an observation, it leaves this little symbol, this little bookmark with the stars around it. So you can tell who typed in manually an identification and who used the algorithm. So this is another case study, if you will, another example of how this can work with other internet tools to provide um, a lot of options for people. So in this case, I I heard a podcast where this fellow, I forget his real last name, but his username James Bird Tour Asia, and um, I was looking for a photo. They found a rare mountain bird in Malaysia, and he said he'd uploaded it to iNaturalist. So after the podcast, I found his account to look at the photo of the bird, and I saw that he also uploaded this photo of a plant in the African violet family. That's what this bottom part is. So it's saying it's genus Codonobia. That's terrible. I should have practiced this. Um, so genus Codo and family Gesneriaceae, which is the Gesneria or the African violet family. So he uploaded this photo, and I thought, wow, that's a very cool plant. Let's see if we can figure this out. So there's another website. So for insects and stuff in North America, Bug Guide is a great resource. For all kinds of um, plants and animals around the world, GBIF, the Global Biodiversity Information Facility, is an aggregator. So it takes data from iNaturalist and from herbaria and from museums and from other websites that do community science, and it puts it all in one place. So if you go there and you search a plant genus, for example, and we go to the next page, so it says, oh, is this the one you mean? So there's the genus, the author, and the taxonomy. And so if we go to occurrences, in this case, we search the occurrences, then it shows me a map. I was just trying to squeeze this together so you saw both parts, but the little map on the far right side, you blow that up essentially to this map with the individual points. And then you can draw a circle or a square on that map or zoom in, find the point that you're looking for, which is close to, in this case, another observation on iNaturalist. And what I found looking at that were some herbaria sheets of of different plants from this mountain region of Taman Negara National Park in Malaysia. So I knew nothing about this plant, but I thought, oh, it's a very pretty looking flower, interesting plant, and unique location near the top of a mountain in tropical Malaysia. So let me see if I can figure this out. And you can see that in the right side of this screenshot, the orange square, that's the iNaturalist observation. And the little red dot to the right of it, that's the point, the locality sourced from GBIF of where the type specimen was collected for this species. So it's proximity, and by looking at the herbaria sheet and the the descriptions of it, was able to say, okay, I think this is the species and provide that identification. So that's all stuff. Those are all web examples. There are also smartphone apps. Right now, there's actually two iPhone apps, a classic app or the one soon to be antiquated iPhone app, and then a newer one that they're working on called iNaturalist Next. And so that's been in development for a while. And then the Android app is just called iNaturalist. So this is a bit of the workflow if you are out on a hike taking pictures of plants. So on the right side, this is a screenshot of my phone's um, gallery, my camera roll from one day walking around looking at plants in New Mexico. And on the left side, options in the new iPhone iNaturalist Next app. So you're able to upload audio or record audio in the app. You're able to upload a photo from your phone, use the camera in the app to take photos, or upload a record without a photo or a sound. So you just put in what you heard and the date and the time, but it it won't reach what's called research grade because it doesn't have the additional supporting evidence of a photo or an audio file. So in this case, if I open the app and I click the upload photo icon, it'll take me to my phone's camera roll. I can select one or multiple photos at a time to upload. And here, if I select a photo of a cactus, then it will give me ID suggestions. So in this case, the first one it came back with was Echinocactus horizontaloneus, which is also called Turk's head, uh, devil's claw, eagle claw. The spines are very fierce. That's what you're supposed to get from the names. So that was the top suggestion. And you can see that there's 5 little dots below the name, which is kind of their showing the confidence of their algorithm in that photo match. And then below it, there are other suggestions, but if you were to click on the eye, the info button, you would see that the Gymnocalycium is from South America and the Coryphantha is probably from Mexico. And then the other one is also from the area where I am, the Texensis horse crippler cactus, but the shape of the cactus is and the color of the cactus is different. So this was the correct name. I already knew it when I was uploading it, but if you don't know it, this is how it suggests things to you. And then to finish uploading your observation, if you do it on your phone and you have allowed your phone to record GPS coordinates with your photos, then it'll automatically populate the location field, which is the second one down below the photo. It's going to take the metadata from your photo with the time and the date and the identification which you've just selected. And then there's a few additional options at the bottom. For example, if you want to obscure your location, so let's say you found a rare orchid and you don't want someone else to come and dig it up, you could obscure your location. And so you know the real location, but everyone else on the platform only sees essentially a bubble of in your general vicinity. If you take a photo in a garden, you can mark the organism, in this case a plant, as cultivated or not wild instead of wild. And then you can also type in additional notes, field notes, or questions or comments. If you do it on the website, it's going to look a bit like on the right. You drag in a photo, and it just provides 4 boxes for the ID. And if you click in there, it'll also give you the algorithm suggestions, and then also the metadata for the photo with the date and the time. Then you have to either, if your photo is tagged with the location, it will import that, or you can manually select one on a map, and then again, notes. So I talked a little bit about GBIF and how I use that to identify a plant. The once observations are confirmed on iNaturalist, they end up on GBIF as well. And they're also available to anyone in the world who wants to look at them. So I found some papers that where they're talking about iNaturalist or analyzing iNaturalist data just as examples. So in this case, this a couple of people from Australia, and they were talking about how There's over 36,000 species. In 2021, over 36,000 species have been uploaded and identified from Australia alone. This was one of the figures from their paper, which I thought was really good. I'll just let you read that for a minute while I drink some water. And this is another paper, also from 2021. People were busy during the pandemic. And so in this case, they actually they looked at biases in the iNaturalist data. So what plants or animals are people more likely to upload? Where are they? And how that influences the data. So now we've gone through all the slides actually, and we have time for questions as well as clicking around the website so I can show you some of these things in person. So show it. I'm going to go to the chat here. Let's see. Okay, so we have the projects question. We have a quality check for IDs. That's also a good question. And what is my iNaturalist name? I can show you all that. And how does it work with Budburst? I'm not sure on that, Maggie. I think you could probably use iNaturalist to upload phenology information. I can show a bit of the phenology part of the website as well. So let's go to the website, and I'm going to silence my phone. Okay, so
Elliot, I'm seeing a black rectangle on your
yes,
screen.
I will fix
Okay.
the screen
Okay.
sharing. Share. Okay. How about now?
It's still there.
Oh, just in the middle?
Yes.
Okay, that's the meeting chat. I have it visible on my screen. I'll close it. So, well, if I move it to the side, is it okay right now? I'm just reading the chat still.
Yeah, it's covering the lower left portion of your or lower right portion of your screen right now.
Yeah. Okay.
Yeah, I don't know if you want to minimize it. It might
Yeah,
take
I
care of the
will.
problem.
So, all right. So we're gonna start with the home screen and go from here. So this is what it looks like on the website, and the majority of the functionality is on the website. The apps are obviously convenient if you're taking photos on your phone. You want that portability, also the ability to record like bird songs or squirrel calls in the app is very nice. But you can do a lot more and a lot faster on the website. So what is my iNaturalist username? It's right here, eGordon88. When I was signing up, I tried eGordon. I tried eGordon1. Both of those were taken. So I was like, okay, what number do I want to try next? 88. Great. That was open. It's as simple as that. And we're just we'll go through these tabs here. Projects is over here. So when you're using the website, if you have interacted with other users, close this bar again. Okay. When you're interacting with other users, you'll get updates from things that happen on the main dashboard. So these are all other people's observations where I added in this case, a high level identification, than someone else has come through and added a more refined one recently. So I've gotten updates about that. You'll also see updates on your own content if it loads. Okay, there we go. So for example, today, someone, in this case, she's a BLM botanist in New Mexico, looked at one of my observations, one of my uploads from 2021, where I had labeled this plant in the photo as Namahispida. And this person said, no, it's actually Tequilia. So I retracted my ID and added one that was not disagreeing anymore. But that was an example of just what kind of updates, what kind of notifications you can get when you're using it. So if we go next to profile, a lot of people do not fill out the profile section on the website. If you use it a lot, or if you just want people to know who you are, I think it's great. I use the website a lot. I have curator privileges, which I can talk a little more about. So I give information about myself and what I do and who I am here so that people know that I'm not just some anonymous username on the website. This is here, these are the statistics for my account. So it shows observations, identifications, annotations, journals, lists, and followers. When you follow someone or when someone follows you, you see their observations pop up in your dashboard. So that in some cases, people upload a lot of things and they clog your entire dashboard. And sometimes you unfollow them even though you still like the person, but it just gets in the way. And then you can also show off favorite taxa on your profile. So in this case, I've selected a manatee, a native bee, Gila monster, a cactus, a pollen wasp, the New Mexico state butterfly, beetles, and these very cool moths that look like wasps. I mean, you'd never know it, right? Look at this. Look how good this mimic is. These are tropical moths that have evolved to to mimic paper wasps, basically social wasps in Central America. And I never would have known about it if it weren't for Inaturalist. There's so many cool things to find out there. This is one that is mimicking a sawfly, which is a on the other branch of the wasp tree. Anyway, we'll move on with the tour and go back to the top here. So observations, when you start uploading things, or if you've already been uploading things, you probably know you can see all the things that you're uploading, and there's a map view. So in this case, this is all the places in the world that I've uploaded observations from Alaska to Hawaii to Peru to over here in Israel and Spain and Norway. And for any species or for yourself on the map, you can draw a little circle or a rectangle and say, oh, what did I see in Boston? Oh, I saw some bees and a fly visiting witch alder on I think that was on the Harvard campus, and some other random things that I saw walking around the city. Now, if we go to the next tab here, this one is edit observations, is a very niche tab. It allows you to select and change properties across multiple observations. That's the main goal of this tab. So it's not something that most people use a lot. But if you upload a bunch of things and then you realize that all of your days were wrong because your camera got reset or something, then you could select multiple observations and edit them in that way. And it's also just another way to see observe where your observations are on the map. The calendar feature, this one is one of my favorites because it shows you not only when you uploaded, but also if you click on this. So for example, if I click on this day here where it says 64 observations and the circle is kind of a darker gray, it's going to tell me that on March 24th, I saw 45 taxa. So I saw 43 insect species, one spider, and one plant. And it tells me where those observations were. And then also tells me which ones that I hadn't seen before that day on Inaturalist. So in this case, it was the first time that I saw anything in this moth genus and this moth species and this moth genus and this moth species and so on. And it also gives you either a grid view of your photos or a list view or again another map view of where you were on that day. So it's a great way to summarize what you saw on any given day and also what was new for you on any given day. In this case, you can tell everything except for the one plant were nocturnal observations, which is a lot of fun. I highly recommend looking out at your porch lights or setting up a black light in the summer to look for moths and other nocturnal animals. As we're moving along the tabs, these are IDs that you make for other people, which can be ones where you agree with someone, you disagree, or you're not sure. And in this case, it's just listed based on your most recent identification and going backwards. So next we get to lists, which is a fun way to look at. You can make your own lists, or there's a life list feature. So if, for example, you want to summarize on iNaturalist, let's say I want to see what corals have I seen. So what animals, in this case, in the phyla Cnidaria or Cnidaria, however it's pronounced. So I've seen some star corals, or I've uploaded some star corals, some anemones, some brain corals, some fan corals, and Corky Sea Finger. What a name! You can do all kinds of exploring and filtering in the dynamic life list to see. So this is what I have seen, and then if I click here, unobserved, that's everything that other people have seen on iNaturalist that I have not. So 1,923 species of um, jellyfish and corals and anemones that I still have to go out there and find and photograph. Now we have just a few left here, and we'll get to one of Maggie's questions. So journal is essentially your own personal blog. You can do almost anything with it. Here, I posted that we would have a plant salvage opportunity near Albuquerque, and then I tagged a bunch of people to notify them to invite them. but you can also use it for informational purposes. So if I go here, I've assembled a list of resources for identifying penstemon that are print resources like field guide. So Flora of Colorado by Jennifer Ackerfield and some other ones. And see here, someone commented, they said, oh, you should add this key to the genus to the list. And so I did. And then here's someone else commented on it and suggested another book on the penstemon subject. So it's available. Everything here is available to anyone on the internet. So anything you put on iNaturalist can come up in a Google search. So you don't want to put anything that you want to keep private, but people who are not logged into the website cannot interact with your content. Here's one other thing I use the journal for. So in this case, the American Penstemon Society, courtesy of David Bleakley, published a key to the penstemon New Mexico, and I reproduced it on the iNaturalist website. So if people were asking how to identify something, I could link them to it very easily. And so there's formatting that you can embed in these journal posts that allow you to, in this case, create a dichotomous key out of it. Plus this nice table at the top with all the species in New Mexico. So we only have a couple tabs left. We have favorites, and you can mark any observation as a favorite. It can be your own observation. It can be someone else's. This tab is going to be kind of slow to load, so I should go ahead and load projects as well. Projects are a social aspect of iNaturalist and also a data collecting aspect of iNaturalist. There are two kinds of projects. So if I click on this one, Colorado Native Bee, any one that uploads a bee on iNaturalist, so any bee in Colorado on iNaturalist will automatically become part of this project. You don't have to join the project or specifically tag your bee with the project. This is a type of project called a collection project. It automatically collects any valid, any qualified observation. Now they should have put in something to exclude honeybees because they're not native, but you could filter that out in the data afterwards. And if you go to stats here, you can see what species have been uploaded, how many identifications have been provided to the observation in the project, and then a snapshot of some of the photos that are within the project. The other type of project is called a traditional project, and I think this is an example. Yes. So this is one called ground nesting bees. I believe the people who are running it are at Cornell. And yes, researchers at Cornell. So in this case, if you see a bee's nest in the ground, you need to be a member of the project, and then you can, on your own observation, you can add it to this project. But you have to go, it has that additional filtering step because there's no natural way within the iNaturalist platform to filter out which observations are nesting bees and which are foraging bees. So that's another kind of project. And anyone can see the data from this project. I could right here, I could export all of the observations and get a list of all of the bees and where those observations are from and what species they are. And here are some people who have, here are some photos of bees in the ground. Like here we have a cellophane bee sitting in a hole in New York. And this person added a lot of notes. Notes are optional. You can add a lot of notes or very few notes. So, okay, the favorites loaded. So favorites, again, you can favorite anything for any reason. Here's an example. I favorited this moth. I marked it as a favorite because I saw it and I thought, wow, that is a cool looking moth. So green. It's clearly got some kind of camouflage for lichen or leaves. And this, I think this is in Madagascar. Yes, it's in Madagascar. And so if I come here and I click, there's two ways you can access the computer vision algorithm on the website. If I click suggest an identification and I click in the species box, then it's automatically going to provide me a list here. I have a web application, a supplemental bit of code that someone in the iNaturalist community produced that gives me a color code here that tells me essentially the confidence of the algorithm. So they did some kind of programming to that it extracts that value from behind the scenes. In the normal view, you just wouldn't have that color shading. You can
Is
also
so Elliot, is that because you're a curator?
no that's available to anyone. In this case, it's um let's see, how would I find tools? Yeah, I'd have to find extensions. There we go. Manage. So it's called the iNaturalist Enhancement Suite, and so anyone can add that to their Chrome browser, and it provides a few extra settings, a few extra visuals in the browser mode. The other way that you can use the computer vision algorithm is if you click compare here. So now there's multiple filters. So if we can filter by place and we can see all observations of moths. So Lepidoptera, all Lepidoptera in Madagascar. And this is just from the most common to the least common. So I'm not going to go through that whole list. But if I change up here to visually similar, now it's going to run it through the algorithm. And now it says, oh, we think it might be this one, or it might be this one, or it might be this one. And so you can go through and do this with a plant or an animal. And you can look at the photos of already existing ones and the photo of yours or someone else's and say, oh, that doesn't look right. Or, oh, that does look right. And then you can always click on one of them, Saurus malacca, and I can come in here and I can see the taxonomy of it. And then on the map, if I zoom out, I can see where other observations of it are. So in this case, this is an Indo-Pacific moth, probably native to Australia, but maybe also in South Asia. So it could be in Madagascar, but it's maybe unlikely. But it's purely matching you here based on the computer vision algorithm, the machine learning and visual similarity of the existing photos and the current photo. So it's not giving you any kind of assurance of the taxonomy itself. If we, well, actually, I can show you something here. So if I go to upload and I choose a file from my computer, if this doesn't go too slow. So let's see. Let's go photos here. So these are the same photos that I took that other night while looking at moths. So if I load one in here, so now the photo is visible here. Here's my photo of a moth. And so if I click here, this is going to do the same suggestion. So it's saying that we're pretty sure it's in this genus. We have a green, so we know the confidence is relatively high. So it gives us a genus as we're pretty sure, and then it gives suggestions. And you can see the top one is in the same genus as the genus suggested, but it's got that additional species with it. So you can upload any photo at any level you want. So if I don't trust this, if I say I can't really tell myself that it's a heteronassa moth, I can just say moth. And then it would say, oh, do you want moth bean? No, I want Lepidoptera moths. And I can upload it as moths. And then anyone who's looking at photos of moths can come along and give me an ID suggestion. But if I do think that the computer might be right, I can click on that. And then I can put in my location and finish uploading it. So I'm going to just close that out for now, but just a sampling of that. We had a question about the quality check. So if I go to the blog, sometimes, all right, there we go. So I'm going to try to find this. In real time. Okay, here we are. This was last year. They did an observation accuracy experiment on the website. So they sent people a list of observations that they hadn't already looked at. And they said, go through this list and on your own merit, judge whether you think add your identification. So if you think the existing one is correct, agree with it. If you think it's wrong, disagree with it. And then they went through, and again, not everyone is going to be. So here, for example, we're assuming that candidate validators can perfectly validate observation as correct, incorrect, or uncertain. So obviously not everyone can identify everything, but they tried to, in this experiment, have a large enough pool of people participate to make it worthwhile. And so they said that in experiment 1, 95% of the research grade observations were judged to be correct. So that means about 5% were either uncertain or incorrect and fixed. And then in the second one, that went up a little bit, but you can see in the other category, needs ID. So research grade means that 2 people have already agreed on the observation, on the identity of whatever's been uploaded. Needs ID means it's sitting out there waiting for someone to identify it. So in that case, about 80% of the identifications were already correct, and then the other 20% needed improvement. So there's been other studies analyzing the accuracy of inaturalist identifications, and people generally come back to say it's in this 90-95% range of correct, and the ease of use of the data, the accessibility, the downloadability, and the ability to correct it makes it a lot more accessible to researchers than a museum collection where either you have to travel there or get a loan. And if there's an herbaria record somewhere and you can't go and validate that voucher, and you can't key it out for yourself, you either have to believe that point or ignore it from your data. But on iNaturalist, if there's something questionable looking, you can go and assess it yourself and change the identification. So if I
Elliot, let me ask you a couple of follow-up questions here.
yeah, I was trying to find the chat again.
Yeah,
Yes,
well,
go ahead.
so while you're doing that, because a few things have come in, but not a lot. So like you showed us before, if my memory was like, right, so somebody from the BLM just more recently changed the ID on a plant that I think you said was from 2021.
Mhm.
So to me, that speaks to what, what you're saying about the updatability, maybe the dynamism of this, because, it could be everything from, and some expert who wasn't available before, comes online and happens to find something and can correct an ID, right? We know there are taxonomic changes. So, a lot of different things can happen over time. And I guess the beauty of this is that, it's not fixed data and information, that it is updatable. I mean, I suppose even if you, from like the study you have on the screen, I mean, even if you have a couple of experts who agree with an ID, that still doesn't mean that in the future somebody else might not come along and still say, oh no, actually you got it wrong. It's, I think it's this.
Yeah. So yeah, absolutely. It's always the database is always open to change and improve. And in terms of taxonomic changes, I want to see if I have any mesquite observations that are old enough to have been changed taxonomically. Okay, yes. So in this case, a big paper happened um, in the last 5 years and moved all the mesquites in New Mexico from the genus Prosopis into either Neltuma or Stromba carpa, I think is the other one. So automatically in the iNaturalist database, here's the citation. So 2022, disintegration of the genus Prosopis. Disintegration. They broke up the genus into 6 separate genera. Um, now, I think they could have just made them subgenre, not made everyone learn new names, but that's discussion for another time. So what happened here is automatically in iNaturalist, in the backbone, any identification and any observation of Prosopis glandulosa, the name was changed to Neltuma glandulosa. So it was automatically updated based on the paper and based on following Plants of the World online, the taxonomic framework for all vascular plants.
So I have one other question for you. And so it's more plant specific, right? Because many people on here are interested in plants, so hopefully not exclusively. Any pointers, tips on taking photos
Mm-hmm.
that can really help? I mean, whether it's the algorithm or just people identifying a species. I mean, it's one thing, and you have some beautiful, macro photos of, of a stationary moth in with a lot of detail. But of course, plants, right? There can be bark, leaves, flowers. So what are your recommendations?
Yeah. So we'll take penstemon for example. So this is another part of the website. I hadn't showed it yet. It's called Identify. And so it's a way to easily browse. The default is 30 at a time observations of other people. So here someone has uploaded a basic leaf photo. Um, in this case, I know this is not a penstemon because penstemon leaves are always opposite. And even in the rosette stage, they don't cluster like this. And I think based on the leaf pairs, which we can kind of see here, it's probably a Boraginaceae. So I can add an ID and I can say, I think it's maybe Asteraceae, question mark. And so now you'll see in the top left there, when I hit enter, it's going to change from Penstemon jamesii to the shared ancestor of Penstemon and Boraginaceae, which is all dicots. But we want to find a good one. Okay, well, here's something else on Inaturalist. Lots of people upload photos for lots of reasons. I upload photos from my garden. There's no problem with that. But iNaturalist data quality asks us to mark photos from gardens as cultivated if that plant is being grown there. And based on this penstemon in this setting with gravel and a walkway and stuff, it seems most likely that it's probably cultivated. And then I'll go ahead and add an ID on it and let them know that I think it is probably Penstemon radii or superbus. But we want to find a good one. Okay, so here's for a penstemon, this is great. So we have flower and we have leaf. Seeing the leaves, always good for a plant. The more photos, really, the answer is the more photos, the better. You don't always have time to stand there and take 5 different angles of a plant, but if you can show at least the flowers and the leaves, that's great. We have with penstemon, being able to see the opening of the flower is always helpful. And being able to see if there's hairs on the corolla, inside or outside, being able to see what the anthers or the stamina look like. These are all really good things. So I can, in this case, confirm that it is Scarlet Bugler or Penstemon barbatus. Close this panel again. So if I want to, I can type out Penstemon barbatus. Or I can type out Scarlet Bugler. In this case, Scarlet Bugler is also the common name of a California penstemon. But we can also, on the website, this doesn't work in the app yet, we can use shorthand. So I can just type PENS space BA, and then it's going to match the letters of those words without me having to remember the whole name. But let's go to Chaffee County instead and look at some random things and see if we have any interesting examples of things uploaded recently. So this is by default that needs ID category. If I include research grade, which I mentioned briefly, so something that is research grade, you can see here, the first person uploaded a photo of a marmot, the second person agreed with it. And so 2 agreements plus an accurate location and time and date meets the criteria for research grade on iNaturalist. And then that observation can then be shared with GBIF and be put into that database. So let's look at plants that are research grade and see if we can find any that are wrong. Here we've got a mountain mahogany. We've got a claret cup cactus, a prickly pear, a pasque flower. Oh, look at that. March 28th, someone saw a blooming pasque flower near Nathrop. An alpine draba. Another draba. All right. So far, everything looks identified correctly. zooming through here to try and find something that might be wrong, but so far no luck. Um, all right. Well, it looks like everything is well under control recently. So, all right. So let's go back to the needs ID observations, the ones that are don't yet have two agreements on them. So here's a Menzelia. It was uploaded 2 days ago, and what we have is looks like some after dark photos. We have some dry capsules, and we have some dry leaves and stuff. So I know this is a Menzelia based on the capsules, but if I want to see what other what Menzelias have been seen in Chappie County on Inaturalist. So here in the info, and then we go to suggestions, we can now see that Menzelia multiflora is the most common, Rusbii, albicollis, decapetala, Cineata nuda, and speciosa. So there's a lot of options, and probably just from the seeds itself, we can't really tell which is which. So we're going to move on to something more grass. Those are too hard. Cheatgrass, that is definitely cheatgrass, even though it's a blurry photo, so we can hit agree. And now up here, it's going to change from needs ID to research grade. A thistle, I'm not going to get in that right now. All right. Let's see. An interesting one. All right. So, anyone who's listening can speak up. What do you think of a distant tree photo with kind of some yellowish stems? And it looks like if we zoom in, it was right along the river.
Cottonwood.
Cottonwood, exactly. So I can hit ID here, hit add an ID, and then type cottonwood, and then I can hit enter. And so now up here it's changed from willow family to cottonwood. Um, a lodgepole pine. All right. So here's an example. So this first person uploaded it, and see they used the computer vision algorithm with this little icon here. And so they uploaded it. They were suggested by the algorithm that it was a Selaginella. Margaret came along and said, "This looks like a real moss to me, no Selaginella." So Selaginella, despite the name, um, spike moss, I think resurrection fern, they're not real mosses. So in this case, the common parent taxonomically of Selaginella and the moss is all plants. So right now I can hit agree with Margaret. And then if another person comes along or the first person changes their ID, then it would update in the system from being plants to mosses. So someone wants to know what kind of tree this is. We have another cottonwood, but let's say we can't remember which type of cottonwood we have. We can again go here to the suggestions, and we can see in the genus of cottonwoods, we have aspen, we have narrowleaf, we have Fremont and eastern and poplars and black cottonwood. Now, some of those may be wrong. That's a story for another time on how you go in and find wrong observations and change them. But in this case, we're pretty sure it's a narrow leaf cottonwood. So we can hit select here. That saves us the typing step. And then now it's going to update. So now I have the leading identification, so to speak, and then the other ones are considered improving based on starting at 0 and getting us down to species. And then, all right, we talked about, Maggie asked about bud burst. We can annotate the phenology of plants. So in this case, I see green leaves. So right here under leaves, I'm going to hit green leaves. I do not see any flowers or fruits or seeds. If we look around, no, I don't see any. So I'm going to hit no flowers or fruits. So now, if we go to the species page, which you can get to by clicking on the name here, you should be able to see the species page now. So this is the view that you can see on iNaturalist. So it has some photos, and we can look through the main photos of what people have uploaded. So there's a bud, and there's a flower. I think that's a male catkin. And then we have some leaves, and some more leaves, and some more leaves. And anyone can change these photos, or we can look at more photos. We can look at the map of all the places that narrowleaf cottonwood has been recorded on iNaturalist, with those other dots, those small dots, again, being from GBIF herbaria. So maybe those are wrong, or maybe those are planted. But if we want to see phenology, over here under charts, first we have seasonality. So this is when are people uploading observations of Populus angustifolia. So it is most commonly uploaded in June, no surprise. And then still fairly common throughout the summer, but we can see it drops way off into the winter on both ends. Now we can see historically. So in the pale gray, those are total observations, and the green is just research grade. So it's split up that way. And we can see that if we look at the pale gray in the background, if you can squint, it is going up over time, rising year over year. The research grade is actually fairly stable for the last four years. And then if we go to sex, so obviously with cottonwoods, we can have male flowers and female flowers. So if we seed in bloom, we can put a sex on the photo. There are only a few male annotations. I see one in September, and 2 cannot be determined in June. So people aren't really using that for this species. And then we go to flowers and fruits. I'm going to hide no annotations so that the scale looks better. There we go. So when are people seeing it in flower? So flower is the blue line here. No, sorry, flower is the red. There we go. So the only 3 people, and you can go in and help with this because anyone can go back and show that anyone can annotate. Only 3 people have annotated flowering Populus angustifolia in iNaturalist. And then a few people have, and then most of the green is no flowers or fruit, so just absence. And then for leaves, red is going to be no leaves, so dormant in the winter. Blue is going to be breaking bud. And then green is going to be with green leaves. And then we have colored leaves for the fall. And that can be any kind of drought condition or deciduous plants. So I'm going to, before we run out of time, I'm going to go back here and click view all observations. I'm going to, on my account, it's set up to automatically filter for New Mexico. So we're going to leave that. And then here under filters at the top, we're going to click identify. That's going to take us back to the identify portal. And it's loading up. And so now I'm going to filter again, filter, and I want to see any without annotations of leaves. Oh, let's do flowers without flower annotation. And so we're going to look at all of the observations, not just needs ID, all the observations and any without flowers annotated. And then we're going to go one more filter. I'm going to go to the month of May and June. So we're more likely to see ones with active flowers. So now I want to find one with flowers. Um, let's see if we can get lucky without taking too much time here. So gonna go to the next page. Oh, wait, that one probably had. Let's see. Those are leaves, leaves, leaves. Sorry, just zooming through here, trying to find some catkins. Ah, here we go. There we go. So we have some seeds here. Great. And then we can see behind that the tree has full of green leaves. So we're going to annotate this as having fruit or seeds, having green leaves, and obviously because there are seeds present, we can say this is female. So we've just added one annotation. This is someone else's observation, RBI underscore consultants.
In La Plata County. So,
Yeah,
so I think so a takeaway from this, like going back to the whole theme of citizen science, is that, not only can you be uploading your observations and helping to, identify maybe other people's observations, right? You could be asking people to help identify your information. But at the same time, if you're knowledgeable and like Elliot apparently have nothing better to do with themselves, you can go through here and query all sorts of things and decide that you want to improve the total database because for example here you're going to add some annotations and that's going to improve the phonology data. Just people are looking for some sort of reference, right? Because I imagine then that feeds back into algorithms as well, because then if somebody produces a photo and it happens to be a fruit, then the database can actually pull from things that now have been identified as fruits.
yes, absolutely. Absolutely. I wanted to address one other thing. Maggie had commented that there are
Exactly. You can
some
be a couch surfing community scientist. Sorry, Elliot.
yeah, yeah. So Kurt here is a member of the Native Plant Society and an iNaturalist user. So I just wanted to highlight if he had put his so I think sorry, just to go back. I think if I search for my full name, yes, so it comes up here. Oh, look, there's another Elliot Gordon. Oh no. Um, but in this case, Kurt has not associated his full name with his profile. So you'd have to know if you search, just say his last name in, oh, so you have to know his actual username to find his. But if we want to see what has Kurt seen, we can find his name. We can go to his observations, and we can see if we go to list view here. These are all from. So at the end of March, about a week ago, in Colorado Springs, he uploaded Pascflower, um, Astragalus, Star Lily, another Pascflower, and oh, and he uploaded an old observation here. So we can help out Kurt because he tried to. So this happens. Let's say you're trying to upload something and your identification disappears. Sometimes it shows up as a placeholder, Lathyrus polymorphus incanus. So if I just copied this here on the web and I paste it in here, and now we can add that identification for him and I will say your ID was a, a placeholder. So, and then as our citizen science, we can go ahead and annotate his observation. one more time. So we see flowers. These look like closed flower buds to me. And then I also see green leaves. Do we see any fruits forming? I do not. So I'm going to go and here and say we have flower buds. We also have flowers and we have green leaves. And you can see here that his observations automatically go to a project that Ackerfield set up called Colorado Flora. All 940,000 plant observations in the state are collected here. So,
That's amazing. All right, Elliot, I'm going to stop you here.
yes, I know
Maggie
we were
had
running out of
Maggie
time.
had I'll say sort of picayune question before. She had a taxonomic question before. I saw in the chat she wanted to know explanation the difference between Opuntia polyacantha versus mycorrhiza.
Okay.
Elliot, of your list, that was the only one where I was like, well, that could be potentially wrong of the plants in Chaffee County.
So if you can catch them after flowering, macrorrhiza has a fleshy fruit. It will be green and then I don't think they really turn deep red, but they'll turn kind of a pink. Polyacantha is one of the dry fruited puntias. So it never has the fruit. It basically just looks like a shrunken bud, and then if you look later in the year, it's just gonna fade to brown. Um, Generally, polyacantha is gonna have a lot more spines. This is something that Donnie and Don Barnett, authors of Cactus of Colorado, could talk a lot more about. But generally, polyacantha is a lot hairier looking or shaggier looking because lots of small white spines. Um, but really, yeah, if you can catch it in flower or in fruit, you'll see the difference between the wet fruits or the fleshy fruits that are eaten, that are meant to be eaten versus the dry fruits that just kind of split open and spill their seeds out.
So thank you. So do other listeners tonight, anybody have any specific iNaturalist questions for Elliot, based on experience and or any kind of aspirational use? And feel free to unmute yourself. I am struck by the huge amount of information and all the features. So it looks like you could spend a lifetime trying to learn iNaturalist.
Yes, yes, you certainly could.
So again, anybody have any questions for Elliot? And even if you haven't used iNaturalist yet, if you want some ideas of how to get started, Elliot's a great person to provide just some basic pointers here.
Well, no worries. If you think of something later, of course, you can email me through Wendy or as well find me on iNaturalist eGordon88. And there is a messaging feature in the website as well, so you can click on anyone's profile and send them a direct message.
Yeah, and I do see, yeah, some David says, thanks, Elliot. Maggie, I'd love to have trainings classes on using aspects of iNaturalist. And I definitely think as members here, if people do have specific ideas, and actually going back to the community science and the city nature challenge. So, I mean, is that pretty widespread now? I guess maybe a question would be like this. Okay, if you're living in a bigger city, in Colorado or something, that's one thing. Okay, what if you're in Salida or Buena Vista or, some small area? Is it necessarily the case that the City Nature Challenge like applies everywhere?
So last year's City Nature Challenge according to Inaturalist, it looks like, oh, this is the global project that does not include any, it's not part of a city that's taking part. So I can find the numbers. Well, actually, probably if I just Google city nature 2024. Let's see how many people participated. So, past results. In 2024, there were in the 4 days of observing, and then you have a week to upload if you're not done. There were 2.4 million observations from 83,000 people. Now, yes, it is generally aimed at cities, at metro areas, like say Denver or Colorado Springs. But if there's no reason that you can't be active in more rural or suburban areas during that time. Your observations still go into that larger pool. So it's
Okay.
still fun, and a lot of people are active, both observing and identifying during that period. So it's it may be a bit on the early side for Central Colorado.
Yes, right. Yeah, it's a timing
And
issue, right? Sure.
yeah, it's optimized to try and even out equatorial areas and Arctic areas, find some middle ground. But it
Okay.
should still, just look at it as an excuse to get outside on that last weekend of April and look at some plants, find something you've never seen before, and have some fun with it.
And there's one question here, so maybe RJ has a question, and maybe we'll use that to kind of wrap this up. So he or she, actually, I'm not sure. I've only used the iNaturalist app on my phone. Where would you say to begin when you transition to the broader website?
Great question. So it's first of all, it's the same login, the same account on the website and the app. So things can go both ways. If I upload something on the website, I can still see it in the app and get notifications about it once the app refreshes. And if I upload on the app, then I go to the website and annotate it and add it to projects and do all those and participate in all those features. So starting with the website, you want to start with looking at your own observations and as well as the explore feature, which is how you search locationally or taxonomically for observations of any kind. And then if you want to help other people, either with the annotations or the identifications, then you can start looking at that identify part of the website. And and I will say one other thing I didn't address before. If you're on the website, on the right hand side near the top of the main page, there's a little rectangle that says forum at the top. And if you click there, it takes you to the iNaturalist forum, which is a free to use online forum. And you can ask questions there about using the website. They ask that you don't put identification questions there to keep those on the platform itself, but the forum is for questions about how to use the website or how to suggest new features, or keeping up with updates from the staff and the team that runs it.
Okay. And, and based on some of these other comments in the chat about, trainings and classes and so forth, and, and also getting out in the field, I definitely think, whether it's our Arkansas Valley chapter or, anybody anywhere else, certainly there are opportunities to roll the use of iNaturalist into some of our summer botany walks, and maybe this summer as well. If Elliot's up in the Chaffee County area, might get to hit him up for an iNaturalist walk. That might be a fun way to do this.
Absolutely.
Well, barring any other questions, I'm just gonna scroll down there. Let's see. I did not ID in the field. How do you look to see if there are any specific identifications, say in a particular location or part of the state? Like if I wanted to see if there's an antelope brittle bush in the San Luis Valley.
Okay, great. I will share my screen back
Okay.
and show that real quick. So let's go ahead and share. And while we were talking, I've been getting more notifications, so I was just opening some of those up. So we want to look for, all right, this is great. So we're going to go to explore, and I'm going to take off the New Mexico tag here. So in this case, so we said antelope.
The brittle bush.
So we start typing, there's
Yep.
antelope
Yep.
bitter brush or brittle bush.
Yeah.
You know, there's one of the features. Let me show this real quick. Go to the species page. So if you scroll down, and you down here, there's more tabs, and I'm going to go to taxonomy, and I scroll down some more. These are all of the common names that people have put in the system. Any valid common name, if you use it or you know someone uses it, you can add it. You can add a name. So in this case, we've got a bunch of English names. We've got some scientific synonyms, and we also have the Navajo name, Sekinachzish. You know, however you pronounce that. So that makes it easier to search for these things if you don't remember the scientific name. Now let's go back. This is the map of all of the worldwide observations. Just refresh it here. All of the worldwide observations of this Persia species. And sorry, the background map is, there we go. Now it's loaded. So it's seen all over the Western US. Now we want to specifically go into Colorado. I'm going to use this rectangular boundary tool, and I'm going to just draw a little box here. We're going to stay kind of central, south central. And so now we have, and so if I want to go closer to the San Luis Valley, I can drag the box over this way. Or if I want to go further north, I can drag the box up. So let's go in close to the area. So there's Poncha, there's Leadville. So let's close the box in, narrow it down. And so now we have filtered 41 observations.
And you might even need to move that, slide that down a little bit. But that's
then,
that's
so
the essence of it.
the,
You could identify a geographic area.
yeah, and then we can go to the grid view or the list view. If we go to the list view, then we can look at actually where those were seen more closely. And then this is the last thing I'll show. If we click on one, we can actually over here on the right side where it has the observer's name and when it was observed and where, we can hit details and we can copy the GPS. And so you can put that in your phone and go right to that exact spot with an alleged accuracy of 4 meters and refine this plant. So that's one way. There's lots of filtering options, but that's just one example.
Yeah, and Maggie says you can also do this if you want to know what plants you might see on a future hike. You can even find specific trails. I often do this to prepare plant lists for botany hikes.
Yeah, so if we go back here to this box and I take off the filter of Persia and I just change it to all plants, now it's going to show all the plant observations within that box I've drawn.
Oh, that's crazy.
including a species list sorted from most to least. So
and for something like that, would you be able to further refine it by say months? So let's say you want to go on a botany hike in July.
In July. So now these are the 841 species that have been seen in that box in July.
Okay, okay. Well, that's a huge amount of information for
Yeah.
one evening. I know there's been a lot of comments here thanking you for sharing this. And like any of these tools available now, right? It's a matter of playing with it, and the more you play with it, the more you're gonna learn its capabilities and get to sort of manipulate it for your own uses and purposes. Well,
Yes.
Elliot, thank you so much.
Thank you everyone who came.